In this video, we are going to be covering the topics in chapter 17, which has to do with nucleic acids and protein synthesis. First, we are going to be discussing the components of nucleic acids. So, understand that nucleic acids are going to be polymers that are made from what we call nucleotides. So again, nucleic acids are polymers. And remember, a polymer is something that we have the smallest subunit linked to one another. So nucleic acids are polymers of nucleotides. So when it comes to the nucleotide general structure, understand that we're going to have, and I'm just going to highlight them, a nitrogen base, we're going to have a sugar, and we're going to have a phosphate group. If we number the carbon atoms in our nucleotide, notice that carbon 1 is linked to the nitrogen base. So let me write it as an arrow right here. And then the phosphate group is bonded specifically to the hydroxyl group that used to be on carbon 5. So on the 5th carbon is where we have the phosphate group, and on carbon 1, so this is carbon 5, that's where the phosphate group is, and on carbon 1, that's where the nitrogen base is. Understand that when it comes to nucleic acids, there are two types of nucleic acids. The first one is called deoxyribonucleic acid, and the abbreviation for it is called DNA. And we also have ribonucleic acid. In ribonucleic acid, the abbreviation for it is RNA. So as you can see, in DNA, we are going to have nitrogen bases that here, these purine bases are going to be the different types of nitrogen bases. And RNA is going to have the same bases, it's just that one is different. And we're going to look at those in detail in the next few slides. So remember, nucleotides have a sugar, they have a nitrogen base, and they have a phosphate. So when it comes to the bases, understand that these bases are going to be nitrogen-containing bases. And these nitrogen-containing bases we learned previously in chapter 14 that are heterocyclic, amines. And we have two types of them. We have pyrimidines and we have purines. For the purposes of this course, all you need to know is that purine rings have 2 rings in their structure, while a pyrimidine ring have 1 ring in the structure. In addition to that, you should know which ones of the nitrogen bases are purines and which ones are pyrimidines. So as you can see, in DNA, we are going to have the purine bases, meaning the ones that are with the double ring, that are called adenine and guanine. And we're going to have the pyrimidine bases, cytosine and thymine. RNA uses the same purine bases. It's just that when it comes to the pyrimidine bases, the only one that is different is instead of having thymine, what happens is that in RNA, we use uracil. So thymine, as you can see, is not used in RNA. When we look at the structures for them in this slide, this is just illustrating how these structures look like. Now, I am not going to require you guys to know the structures specifically for cytosine, thymine, uracil, adenine, and guanine, but you should know. Which bases are purines and which ones are pyrimidines and which nucleic acid they are found. So that's the information that I require for you guys to know. Now let's move on to the sugar motif found in the nucleic acids. As I mentioned before, RNA is ribonucleic acids. The ribo in the name is because the sugar motif in RNA is ribose. In DNA, remember that this is deoxyribo. So the sugar that is found in DNA is deoxyribose. So if you ask yourself, what is specifically the difference of deoxyribose and ribose, as you can see in the figure in the bottom of the slide, the difference is that at carbon 2, in the deoxyribose, we have a CH2, while in the ribose sugar, we have a CHOH. Now, deoxy means lack of oxygen. And then as you can see, this lack of oxygen in the deoxyribose is going to be at carbon number 2 in the ring system. Now, when we have the sugar bonded to the nitrogen base, that is known as a nucleoside, okay? And the reaction that occurs in order for them to be linked to one another is the removal of water. Remember that all of this is done by enzymatic reactions. So in essence, what is occurring in this reaction is that the H in the nitrogen base and the OH in the sugar motif is going to be linked to one another, and that's where that's where the water is formed. Then the bond itself that is found on carbon 1 that links your sugar motif to the nitrogen base, as you can see, has a specific name. It is called an N-glycosidic bond. It is called N because it's bonded to nitrogen. It is called glycosidic because it's bonded to a sugar. Now, you may ask yourself, what is the difference between a nucleoside and a nucleotide? The difference between a nucleoside and a nucleotide is the presence of that phosphate group on the 5' carbon in the sugar motif. Okay, so understand that when it comes to the nucleotides for RNA and for DNA, they are pretty much the same. The only difference is that when we look at them, remember that the ones in DNA, okay, so I'm just going to make a note of that. When it comes to the nucleotides in RNA, they're going to contain a phosphate group, they're going to contain a ribose sugar, and the nitrogen base. When it comes to DNA, and remember that the nitrogen bases in RNA are going to be adenine, cytosine, guanine, and uracil. When it comes to DNA, remember that they are going to be composed of phosphate. Then we're going to have a deoxyribose. And we're going to have a nitrogen base. In the nitrogen bases that we're going to have in DNA, they're going to be adenine, thymine, guanine, and cytosine. Now, understand that the phosphates, we can have 1, 2, or 3 phosphates. Here in the slides, we're only using 1 phosphate, okay? Now, when we have 1 phosphate, that's why we have the name monophosphate. But note that nucleotides can have 1, 2, or 3 phosphates. If it has 1 phosphate, then that's where you see the prefix mono. If you have 2 phosphates, then we're going to have the prefix di. And if we have 3 phosphates, you're going to have the prefix tri. Okay? Now, as you can see, when it comes to the naming of the nucleotides, you can have a different name representing the nucleoside and the nucleotide. So understand that the names that I put in the corner are specifically for the nitrogen base. When they make nucleosides and nucleotides, these are the names that they obtain. Adenine goes to adenosine. Guanine goes to guanosine. Cytosine goes to cytidine. Uracil goes to uridine. And thymine goes to thymidine. Remember, thymidine is the only one that goes deoxy because it's in DNA, and uridine must have a ribose sugar. So that's why we have those 2 distinct molecules in the bottom.